Good morning. It's great to be here speaking to you today. My name is Francesca Nicholls and I work in an academic lab at Oxford University in Simon Lovestone's lab, and we also work very closely with the Drug Discovery Institute here. Ooh. Is that better? Okay, excellent. Okay. So I'm just going to tell you a little bit about cell models for drug discovery. So I think before I entered science, when somebody said models, I probably would have thought Lego. Unfortunately, that's not what we do much of in the lab, but I'm going to tell you what we mean by cell models and how we use them to look for new drugs. So by cell models, all we really mean is cells in a dish. So they grow along on the plastic surface. We give them medium, which is the nutrient mix that allows them to grow and function normally. And we keep them at 37 degrees at body temperature so that they function as close as possible to cells in the body. To give you a bit of an idea about scale, a cell is about 10 micrometers across. So that means that if you lined up 1,000 cells, the line would be 1 centimeter long. So obviously we do all our work with microscopes and so on. So this is what neurons look like. These kind of bright centers are the cell body, and then they extend out these neurites, which is how they communicate with surrounding cells. So this is a happy, healthy neuron, but in the disease state, they die, and all that's left here is just some cell debris. And what we want to do is find molecules that halt this process. So, but neuronal loss is really what happens at the end of the disease process. So, if possible, we want to look a bit earlier than that. So, one option would be looking when they're, sorry, that hasn't really worked, but when they're just a bit sick. So you can see with that one that it's lost some of its neurites, it's got a bit smaller, and that's something that we can also look for. So to try and catch that process a little bit earlier. But what if they look the same? So that neuron looks the same as the one above, but we don't know whether it's still really functioning as a neuron. How is it communicating with surrounding cells? Because that's the process that really underlies our cognition and underlies the symptoms of dementia, is these neurons being able to effectively communicate with each other. So what we're now able to do in the lab is we insert a protein into our cells, which is light sensitive. So that means that when we shine a light on it, we can get it to send a signal. So it sends a signal to the next cell and then to the next cell. And then we measure the cell at the end. So we can see if that signal has been successfully transmitted, which means that our neurons are functioning properly. So if we look at that in the disease state, we shine the light again and it sends a signal, but it might get stuck. So when we then look at the receiver cell, we don't see any signal there. So that means that we can really look at drugs that are preserving that essential function of neurons, of being able to communicate with each other. But all these models still have a place. So the functional model that I was just describing is, we think, more relevant because we're preserving that function, but we need quite complicated equipment to measure that, and it takes quite a lot of time. So we have to find the right model for each application. And so where do we start from in terms of drugs? And they're in libraries. So drug libraries are very similar to our libraries, to book libraries in a lot of ways, in that they range from small collections of very targeted molecules, so where an academic lab has found a very particular mechanism in the cell that they think is important. We can find a very focused library and do a small number of experiments to see if any drugs targeting that mechanism are useful. And then there's sort of larger but still specialized libraries. So these might be things that, for example, that we know get into the brain easily. So one of the big problems we have with diseases of the brain is that the brain is really well protected, which is usually brilliant because we don't want lots of things getting into the brain, and it helps with other medications, and not a lot have side effects in the brain, which is great. But when we want to target the brain, we have to get past the blood-brain barrier. So there are some libraries that are specially designed that they're all molecules that will be able to get across that barrier. And then we, of course, also have the giant libraries. So these will contain hundreds of thousands or even millions of compounds. And the idea there is that it gives us more chance of finding things that we wouldn't have expected. So if we know of a particular mechanism to look at, that's great. But sometimes we just want to try and see maybe there's something we missed that we didn't know about that would show up in one of these really big screens. So I guess the typical thing we think of cells growing in is a dish like this. But obviously when we're looking at these big libraries, we can't really have thousands of these. It's just unwieldy. So we're able to do a large number of experiments in parallel by using ones like this. So this is called a 96-well plate because it has 96 wells, and that means that we can do 96 experiments at the same time. So these all go into our measuring equipment in one set, and it really helps us to be able to maximize the number of experiments that we can do. And there are ones where you can do even more experiments in parallel as well. So if I now go back to the different models, you can see that the simpler models are going to fit better when we want to look at lots and lots of drugs, and the more complex models are going to fit better when we're looking at a very specific small library. And this comes into play in terms of where we get the cells from as well, because a lot of the time we use rodent cells, but we're now also able to use human cells because patients or healthy individuals will donate a little bit of their skin or hair, and we're now able to turn that into neurons in the dish so that we can study the difference between a patient's nerve cell and a healthy person's nerve cell. So how does this actually work in practice? So we take our 96-well plate, which is full of, well, we can do it a couple of different ways. So as Robin was saying, we can look for symptomatic drugs. So there we would put in sick neurons, and we would look to see if any drugs can improve them. But what we really want to do is find things that alter the disease course. So we want to put in healthy neurons, and we want to add our drugs, and then most of these neurons during the course of our experiment will start to lose their functionality, and we want to find drugs that will preserve that. So we take our drugs from the library and we put them into our 96-well plate, and as you can see, each one is represented 3 times, because in biology we never believe anything unless we've seen it happen at least 3 times. Usually a lot more than that, but we want to be sure that these drugs are going to work every time. So what we hope for is that there'll be a clear difference, and we'll see that there's a few of these wells in which the cells have really maintained their health and their functionality, and then we know that this is the drug that we want to take forward to development. And so there's quite a long pipeline, obviously, and so the academic labs work a lot on finding the mechanisms of disease, and slowly the pipeline goes through to the pharma companies who do the clinical trials and that kind of thing. And what we now are lucky to have in Oxford is a drug discovery institute. And what they do is they're in the middle. They're a bridge between the academic labs and the pharma companies, because they're able to take all these ideas that come from the academic labs and I guess really sift through them and see which are going to be the best targets, the best drugs to pass on to pharma for further development. And they'll have an information stand out in the hallway, so if you want to find out any more about the drug discovery process, then I suggest you go and chat to them. Thank you.